Hello, welcome to the first mini lesson in our Chapter 3 journey. Chapter 3 is broken up into two parts. So part 1 is going to be dealing mainly with just cell biology. We're going to cover the structure of the cell and some of the functions of the cell, like membrane transport, cell division, and protein synthesis. And then the second half of the part 2 of the chapter covers histology, which is tissues. We're going to be taking a look at different types of tissues. So let's go ahead and get started taking a look at the anatomy of a cell. So you have this diagram in your book or something similar to this showing a generalized cell. So not all cells look like this. It's just kind of a nice colorful way to illustrate the general appearances of these features. So I'm not going to go over everything in this particular diagram because we're going to go individually, but I would encourage you, after you've spent some time working on this part 1 of the chapter, that you might go and try to label a picture like this. Maybe try to find a blank one or draw your own cell. That's always a great way to learn. Making sure you're comfortable recognizing what these parts are and what their names are. So, you know, if you can make yourself your own little vocabulary list or get flashcards if you want to do that, or have somebody quiz you, but make sure you're comfortable labeling all of these structures on a picture of a cell. The first part we're going to take a look at is the nucleus. It's the center of the cell. It holds all of the DNA, and it kind of is the boss of the cell. It has all the instructions for everything that the cell is going to do. So the nucleus is what we call membrane bound by an envelope. Okay? And then within this, what's enclosed by this envelope is what we call chromatin, which is basically another word for your DNA. Okay, so your genetic information in this kind of loosely organized state is called chromatin. And in the very center, we have what's called a nucleolus. It means like little nucleus. It is in the nucleolus where ribosomes are kind of assembled. You may not know what a ribosome is yet because it's coming a little bit later, but once we do learn about ribosomes, just know they are put together in the nucleolus, which is inside the nucleus. And then the last feature to take a look at for the nucleus is it has holes in it. It has nuclear pores. There has to be a way to communicate from the DNA inside of the cell to the rest of the organelles, or inside of the nucleus, the DNA inside of the nucleus, to the rest of the structures and organelles outside. So those instructions can't be sealed off and just kind of tight in a sealed off nucleus. So those pores allow for communication from the nucleus out to the cell and from the cell into the nucleus to regulate maybe some of those instructions. The next part that we're going to take a look at is the plasma membrane. So plasma membranes are made up of mostly phospholipids. So you may remember in your chapter 2 section when we're talking about lipids of our major macromolecules, there was one that had a charged head, right? So it had a phosphate group, it had polar properties to those molecules, and then it had these long fatty acid tails that are neutral in charge. So what's unique about these phospholipids are they themselves are polar. So we have a charged end and we have an uncharged end. So what we end up getting are all of the charged ends, or polar heads, pointing towards the watery environment outside of the cell or the watery environment inside of the cell. And all the hydro or the lipid tails, which don't like to mix with water, these guys here, they are all pointing towards each other to make up what's called this bilayer. So we have this 2 layers of lipids. And so on your picture here, it's these gray balls on the outside edge. Because they have that charged property, they're okay to be next to water because water is polar. Remember that was one of our characteristics of water. And so these are happy to be next to water. Yay. This is hydrophobic. They don't like to be next to water. So hydrophobic means they're afraid of water. It's not that they're actually afraid, but they're lipidy. There is no charge here. So they just repel any water. So we use the term, I'm not sure if they use this in this book, but it's called hydrophilic, which means water loving, and hydrophobic, which means water fearing or water hating. So that is, we have this kind of bilayer with all the lipid tails pointing towards each other, and then we have the heads that are okay to be around water. We'll put water, oops, question mark. Yay or no or yay. So embedded also in this lipid bilayer, these big purple blobs are called proteins. So they could be structural proteins like anchors holding the cytoskeleton together. They could be a channel, like this is a channel that could allow things to move into and out of the cell. So the plasma membrane is kind of like this fluidy lipid bilayer that can allow things to move in and out of it, which we'll see in our cell transport function, either through the bilayer itself or through those channel proteins. Okay. Oh, one of the words I wanted to make points here in the notes is it says it's semi-permeable, which means some things can pass through it and other things cannot. And we're going to focus more on that term when we get to the membrane transport section of the functions. With that idea in mind of these plasma membranes, I did want to point out there are some specializations that are found in certain tissues where the cells are right next to each other. And these are called cell junctions. Yeah, membrane junctions. So this first picture here, kind of showing the two purple plasma membranes right up next to each other. And then these little green, they look like staples to me. Right? These are called tight junctions. What they do, they're basically stapling and sealing two neighboring cells together. And we can see that right here. Kind of peeled one of those membranes back. But those tight junctions hold neighboring cells really tight together. The next one listed here are desmosomes. So this is a desmosome down here. Desmosomes are like anchors. So here's one in this picture. So they tightly hold two neighboring cells together. So tight junctions, I know their name is tight, but they're like a seal, like if you do the vacuum pack, right? So you pull down on your little handle and it seals the vacuum or seals the plastic bag closed. That's what tight junctions are. Desmosomes are more like strength. They're anchoring, they're holding cells physically together, not just sealing the membrane. So there's a lot more strength behind desmosomes. And then the last one are gap junctions. And what gap junctions do, they allow cells to communicate to each other. So stuff can move through these pores, these holes, these gaps right here. Okay, so things can move back and forth through those gap junctions. So we have tight junctions, which are like sealing the plasma membranes, desmosomes, which are strength and anchoring, and then gap junctions are communicating. They allow things to move back and forth. They could be ions or hormones or nutrients. Okay, so I brought the picture back of the generalized cell because the next thing in the notes is talking about cytoplasm. And the cytoplasm or cytosol is just a liquidy gel-like material that everything else is floating in. So you have your cytoplasm or cytosol. It's where your organelles and your nutrients and waste products are all just kind of floating around in. The next organelle is our mitochondria. And this I usually try to help students remember it by calling it the mighty mitochondria because this is where ATP is produced. And if you remember from our chemistry chapter, ATP is our energy currency of the cell. This is the molecule that all the cell processes use to get work done, whether that's going to be pumping something or doing cell division or making a protein or making a hormone. They need the power of ATP to do that, and all of that ATP comes from the mitochondria. Okay. The next little structure are called ribosomes. So I have lots of different pictures of ribosomes. Ribosomes are these two-unit structures. They almost act like a sandwich when they go to make a protein. So ribosomes are the site of protein synthesis. What that means is this is where proteins are made. And we're going to get to that whole process. We're going to have a whole little lecture on that all by itself. But usually what they're represented in pictures or diagrams is they're just little dots, right? So you kind of see those little dots there on the endoplasmic reticulum. Each one of these little black circles is representing a ribosome. They're just small little structures that you can see floating around in the cytoplasm, or sometimes you'll see them attached to the endoplasmic reticulum. And here we have the endoplasmic reticulum with some ribosomes attached to it. So the ER for short, and I'm okay with you abbreviating ER in lab or your exams or quizzes. Endoplasmic reticulum, there's two major types. We have what's called the rough endoplasmic reticulum. And the reason why it's called rough is because it has those ribosomes attached to it. It makes it look bumpy. And then we have smooth endoplasmic reticulum, which does not have ribosomes. So that's the major difference structurally is rough ER has ribosomes, smooth ER does not. They do have different functions. So because the rough ER has loaded up with ribosomes, it's a site of a lot of protein synthesis. You're going to have a lot of proteins being made on that rough ER, and those proteins typically go out to be implanted in the membrane, the surface boundary of the cell. Smooth muscle, or sorry, not smooth muscle, smooth ER, smooth endoplasmic reticulum sometimes can be a storage tank. Right, we can see all these little tubes, which would be good for storage of muscle. They store calcium ions. They also can produce lipids and hormones like steroid hormones and cholesterols and things like that. So that's your smooth ER and your rough ER. Next, we have the Golgi apparatus, or sometimes called the Golgi Body. So that's the green blob right here in the middle, kind of sitting right next to the rough ER. They kind of work in tandem. So all of the proteins and things that are made by those ribosomes and the rough endoplasmic reticulum, they get packaged up in these little vesicles. Vesicles like a little bubble, like a transport bubble that carries things from one place to the next. So this little bubble, this vesicle from the rough ER with these proteins in it, goes and binds to the Golgi body. The Golgi body functions to kind of process, do some final tweaking on that protein product, and then it packages it into another vesicle, which is headed towards the outside of the cell, and it will release that process via what's called exocytosis. So a lot of times people think of the Golgi as like the FedEx of the cell. Think shipping and packaging, or packaging and shipping. It receives a package from the rough ER, processes that, maybe adds some sugar molecules or tweaks the protein shape a little bit, packages it up into a new vesicle, and then ships it off to its destination, which could be the surface for secretion. It could be another location in the cell, maybe to the mitochondria or another organelle. Okay. So there's a drawing of it, and then here is the micrograph. We can see the micrograph picture. It's just a whole bunch of folded membranes, and you usually see all these little vesicles coming off, these little bubbles going into and coming out of the Golgi. All right, and the next is our lysosome and peroxisomes. These are small little organelles, and their job is to kind of detoxify the cell. The lysosome is like sometimes the garbage recycling center. So your cell debris, any damaged parts of the cell can go in, fuse with the lysosome, and then be destroyed. There's enzymes and acids and things in there. And then the peroxisome is like a detox, specifically for toxic forms of oxygen, like hydrogen peroxide. So there's enzymes like oxidase that are found within the peroxisome that help to neutralize some of those toxic forms. If you've ever heard of things like free radicals and antioxidants, so your peroxisome is like a built-in antioxidant, helps you get rid of some of those dangerous chemicals that could damage your cell. All right, and then we have our cytoskeleton. So this is what gives cells its shape and structures. A very unique or complex shape, you're going to be seeing a lot more complex cytoskeleton. And centrioles, these are centrioles right here. Centrioles are involved, actually they would be sitting right here, let me change the color, right here in a cell during mitosis. But I included this picture of kind of these chromosomes going like this because all of those red fibers are made up of these microtubules, these guys right here, and they are what help to replicate the cell during mitosis, which we'll be learning about shortly. But the centrioles play a role in that cell division. The other images on the screen are just showing you the diversity and the kind of how complicated some of our cytoskeleton structures can be. All right, so that wraps up the anatomy of a generalized cell. And like I said earlier, make sure you are comfortable labeling a cell, identifying all these parts and pieces, describing their general appearance or their general function. All right, I'll see you next time for some cell function. Bye.